few minutes, see who else wants to show up, and we'll kind of get underway here. Both of them are back here. Maybe all three of them are back here. I mean, I don't know. They're all underneath the chair. Looks like we got one more joining. All right, maybe just added just now. Okay. Great. So I think I started the recording already. It's January 31st, Sunday evening. Looks like we got a good crowd tonight. It's probably getting closer to quiz and first exam, so I suspect that we'll be getting more attendance. The attendance will go up, certainly. Okay, so earlier this week, we really have been talking a lot about, I think I have sound, Colton. Can you guys hear me? Okay.
Yes.
All right, some people can hear me, okay. Sometimes I mute my own box. All right, sounds good. So last week we've been doing a lot of lecture stuff, a lot of biochemistry, a lot of biomolecules, lipids, polar bonds, proteins, let's see, glucose, polysaccharides, monosaccharides. You may have to mute them, Kay. I think that's what I do sometimes. I'm not sure, Colton. You can always type in you could either turn us up on your volume. I'm not sure why it would. You may want, it may be like a, you could reach out to the, I think I forget his name. There's a guy that works these, he's an engineer that kind of monitors this. And you could reach out to him and maybe he has an answer for you. I'm not sure if it's your computer or if it's the Blackboard Collaborate itself. You can go into settings or something. But okay. So does anybody have any excuse me, does anybody have any questions about any biomolecules, like what I just mentioned, polysaccharides, lipids, any lecture material that we can get started on today? Nucleotides? What are we doing in lecture this week? Well, maybe I'll maybe people can't hear me. Does everybody understand biomolecules? Is there soft spots that we need to go over? Okay. While people are thinking about that, then I'll probably just talk about the cellular form and function this week. So okay, so structure kind of yields function. All right. So is there any questions about that? Does anybody have any questions about what Meredith's topic is? Different organelles. If we're doing organelles this week, perhaps what they do, what their purpose is. Are we okay on all that? Usually chapter 2 and chapter 3. Okay. Who do you have for lecture? Kiasha? Depending on who you have for lecture may help out how they study. Normally it's PowerPoint PowerPoint slides. Okay, she's probably for lab. That's probably your lab person. So usually for lab, you would study just the lab manual. Okay, and Dr. Sharp. So we have these spotlight sessions. Great, we have these spotlight sessions. So if you guys have a question about lecture, then I can help you out. All right, Sierra, you're struggling with bonds. All right, cool. We'll talk about bonds. Thank you. We'll talk about bonds here in a minute. Go back to Kiasha's question is that she is the lab person. So for lab, you need to study what's bolded in the lab. You need to study, there's arrows and bolded words in the lab manual. I think we're doing lab 2 this week. And that is something that's, you know, that always we pull questions from. Okay. What kind of chemistry? What kind of chemistry? I know Meredith talked about bonds. We can do polar and nonpolar bonds, or ionic and covalent bonds. I'm assuming that's what we want to target. Kiasha, if you have something that like a specific topic, then I could try to help you out. Okay, well, let's start off with that. Polar and nonpolar bonds. So basically, polar bonds are bonds that can bind water. Okay, they're able to bind oxygen. They're able to bind water. Okay. All right, Karen, that's cool. Just help direct this conversation the way you all want it. Okay, I'm going to start off with bonds and our polar and nonpolar bonds, and then if you want to dive in deeper to ionic and covalent or a different type of bond or proteins, then just text me something, and I'll continue to talk this, you know, as long as we can. So polar bonds are bonds that bind water. Okay, they bind oxygen, they bind hydrogen, and this is usually what takes place in our body. Proteins, glucose, polysaccharides, they can all bind water, so they all have polar bonds. Non-polar bonds, on the other hand, are mostly seen in lipids, fats. Okay, these are like oils. They do not bind water. You know how if we were to take water and oil and shake them together, then they kind of separate out. That is what polar bonds and non-polar bonds do. Excuse me. Polar bonds will bind in the water layer, and then non-polar bonds usually bind themselves. Does that help clear any of that up? Okay. Okay. Cool. Now, do we want to talk about? Okay, good. Thank you. Do we want to talk about ionic and covalent bonds? Did we do that, or hydrogen bonds? Think about, do we cover any of that in the lecture? Okay, all right. All right. So hydrogen bonds basically combine oxygen, nitrogen, or fluorine. All right. Hydrogen bonds are bonds that we can find in between the DNA strands, between the nucleotides. Fairly a strong bond. They are found inside of protein foldings between amino acids. And hydrogen bonds can either bind themselves, or they can bind, you can have a hydrogen and oxygen, hydrogen and nitrogen, or hydrogen and a fluorine. And that basically just works off the electronegativity of the oxygen, the nitrogen, and the fluorine. Now, you might think, well, what is electronegativity? These are elements or atoms that love electrons. Okay, so when you think about it, you have a hydrogen. Hydrogen is usually written as H+, right? Well, it has one electron. And what that means is that it can quickly be stolen by another atom that loves electrons more. All right, and usually if you were to look at the periodic table, on the right-hand side, you have fluorine, nitrogen, and oxygen all next to each other in the top right. And fluorine obviously is the most electronegative of them all. They are the one atom that loves electrons, loves taking electrons more than anything else. And what happens is that when you have hydrogen next to it, it will rob that that electron from hydrogen, and that creates a bond, okay? And they're a sharing bond, so that's a covalent bond, or also what we call a hydrogen bond. All right, now let's talk about covalent because I just introduced that term. That's what electronegativity is, okay? That's the most electronegative atoms is fluorine, and then it's oxygen, and then it's nitrogen. So that's why you have hydrogen bonds to those three atoms. And they can be fairly strong when you have a bunch of them. We see them a lot in DNA structure and protein structure. So we okay on hydrogen bonds and electronegativity? We'll just take this one step at a time. Okay. All right. Thanks, Karen. All right. Okay, Kendra. Sweet. All right. So covalent bonds. Covalent bonds are a bond that shares electrons. Okay, they share electrons between each other. They have what's called a dipole moment. Basically, what that means is you have these little charges. You know, charges is in like between protons, neutrons, and electrons. They create these charges to where they can share electrons together, and they create a bond. Okay, this is different than ionic bonds. Ionic bonds basically can grab it. Ionic bonds basically are bonds that electrons can be given up easily. If you were to look at the left-hand side of the periodic table, you're going to see a lot of metals, what we call metals, or atoms that can quickly become charged, like potassium. Potassium will break up into +1, and lithium is another one, magnesium is one, calcium is one, and they're all on the left-hand side of the periodic table. And these bonds quickly give up their electrons. Now, if you remember, electrons are negative. So if you were to give up an electron, that means that they are becoming positively charged. Now, that first column is what we call the alkali metals, and they give up their electron readily. Like sodium's in there, potassium's in there. There's a few other ones, maybe lithium, I can't remember, beryllium perhaps, and they give up their one electron. Yeah, you may have to find who it is, Kelly, and mute them, I think. Yeah, guys, if you've got some background or your TV on in the background, you may just want to mute your microphone. Okay. All right. So if we have these atoms and they quickly give up their electrons, and they are able to. That's how they bond. Okay, when they give up their electron, that is pretty much their commitment to making a bond. If chloride would take it or another atom would take it, then and you create what we call an ionized bond. That means that they are charged. All right, they're pretty much stealing, like one atom is positive and one atom is negative, and that creates a charged two charged ions together create an ionic bond. So when I say ion, what I'm really meaning is that that's an atom. That was an atom that is charged now. Okay, it's either negatively charged, which is an anion, or it's positively charged and it's a cation. Most atoms can become charged. The only ones that can't become charged are the ones on the far right, which are the inert gases. And the rest of them can be, you can take electrons or you could add electrons to them. Like fluorine loves to have 8 electrons. Now, normally it's 7. So if it steals an 8th electron to fulfill its octet, a total of 8 electrons, and there all of a sudden you would see fluorine being negative. So it would be F negative, which is an anion. Chloride's the same way. Those are the ones are on the far right of the periodic table. They love to steal electrons. So if you stick them with calcium, like potassium fluoride, chances are the fluoride's going to have potassium's 1 electron because it's going to rob it. And then that gives potassium a +1 and fluorine a -1. So that's kind of like when you have ionic bonds, we're not completely sharing, we're more like stealing the electron, and we create ions. So what do we think about all that? That's kind of a little bit more involved because I threw a bunch of vocabulary words at you. And I'm assuming you looked in that, well, not you, I'm assuming that we covered in lecture. Okay. Is there any other chemistry? We did biomolecules earlier this week, and a lot of people seem to like that. But like nucleotides and proteins and yeah, okay. Yeah, that's right, Kelly. So each we have, well, I'm trying to think what we call them. Electron layers, electron bands. Yes, that's when you can have 2 electrons in the rings. Yeah, electron rings. Okay. So yes, you would have 2 in the first one, and then you'd have 4, and then 6. I think it's, yeah, I guess it is update. That's more like where they are around the atom. We can never really tell where, if somebody were to ask you what that, I think it's 2, 4, 6. I don't know how far deep down it goes, but you can only have, they have this octet rule too, which basically I think it goes up to 8, honestly, where the outer electron ring is also known as the valence ring, and right, the valence ring, and so those are the electrons that get stolen. Okay, the valence shell, that's the electrons that get picked off to create these bonds. Okay, it's not the ones that are closer to the inside. It's the ones that are from the complete outside that get picked off. So they want to satisfy this octet rule, which is 8. And fluorine especially does, because fluorine has 7. So it only needs to get to 8. So it'll rob an electron from, let's say, potassium, who's willing to give up their 1 electron in their valence shell because it's more comfortable without that electron. Does that make sense? Like the ones on the left, the atoms on the left always want to give up either 1 or 2. That's why you have magnesium 2+, calcium 2+, because those guys give up their 2 electrons to go down, to give in their valence shell, to go down to become a cation. It's what they prefer. They were looking for somebody to take their electrons. Okay. So I'm going to let that sink in there for a minute. Make sure we're okay there. Anion, I just put in the chat. That's negative. The cation is always an ion that's charged positively, and then the anion would be negatively charged. So if you have an anion, that means it's taken on an extra electron. Yeah, no problem. It's taken on an extra electron because electrons are negatively charged. So if I'm an element, or if I'm an atom and I take on an extra electron, then I'm going to be an anion. If I were to give an electron, then I've just lost a negative charge. So that makes me positively charged. Therefore, I am a cation. All right. So I think we okay on all that? Is there any other questions that we want to dig into? Does that help clear up some chemistry? Chapter 2 is pretty involved. It probably is one of the hardest chapters. Is there anything else in chapter 2 we want to go over? Okay. Well, let me know, Karen, if we can get even better than that. You just kind of have to tell me what we want to focus on. I'm trying. I haven't seen those lecture notes at all, or I would pull out some topics. How are we on biochemistry? Are we okay on biomolecules? Like, uh, positive, negative feedback is in like endocrinology. Like, are we talking like hormones and how they answer back on each other? Is that what we're talking about, Kiasha? Okay, okay. Well, I'm glad you brought that up because I'm kind of like a baller in that topic. Most of the time, when we have hormones, when we talk about positive and negative feedback, we're talking about three different parts of the human body. All right, we're going to focus on the hypothalamus. We're going to focus on the pituitary gland, and we're also going to focus on what we call the target gland. All right, when I talk about glands are tissues that release hormones. Okay, let's talk about what happens when we get stressed out at school or we get stressed out in life. We're going to talk about cortisol. So what happens is that, and it all begins in the hypothalamus, which is in part of your brain, right? This is the hypothalamus will release a it will release a hormone. I think it's called uh, corticotropin releasing hormone. I don't want you to get tied up on all these vocabulary words, but when you see a hormone called blank releasing hormone, like the key word there is releasing hormone, chances are we're talking about the hypothalamus. That's going to be the hormone that gets released from the hypothalamus. It will release to the hypothalamus and it'll travel down to the pituitary gland, which is at the bottom of your brain. And that will stimulate another gland. And I think if we're talking about cortisol, we're going to talk about adrenocorticotropin releasing hormone or stimulating. I think it's stimulating hormone now. And what happens is that it will release another hormone from the pituitary gland. Now, what happens there is that now that hormone will go systemic in your blood. It'll go everywhere. When I mean systemic, I say it'll go throughout your whole body to go find its target gland, because that means it's going to take another message. And it could either travel to the kidneys, it could travel to the liver, it could travel to the heart. Hormones are pretty much just proteins that travel a long distance to deliver a signal to somewhere else. And in this case, we're going to the adrenal glands on top of the kidneys. The adrenal gland on top of the kidney is a gland that houses cortisol, and it will release cortisol out into the body to carry out various roles. Cortisol carries out a lot of roles. It'll affect your heart. It'll affect your blood sugar levels. It's really not good for you in the long run. Cortisol levels are not good for you in the long run, but it kind of helps your body deal with stress. So what happens is that when this is the negative feedback part, when we have the stimulation of cortisol, we don't want this to continue going, right? I mean, if we have more hypothalamus releasing hormone, that means we have more pituitary stimulating hormone, and that means we get all kinds of this cortisol. I mean, we got like a lot, tons of cortisol being issued out into the body. So what happens is this cortisol will travel back up to the pituitary gland and will feed back. It will pretty much tell the pituitary gland and the hypothalamus gland that enough is enough. So you need to calm down and stop releasing so much releasing and stimulating hormone. So this is how we can control how much cortisol we let out in the body. TSH, thyroid stimulating hormones, another one. In females' reproductive systems, estrogen, follicle stimulating hormone, luteinizing hormone. All these are different hormones that act underneath a negative feedback system. Now, there's only 2 positive feedback systems in the body. All right. And one is, honestly, I can't think of the second one because it's rare. But the big one that is a positive feedback is during childbirth. Yeah, that's right, Meredith. So what happens is that I think it's oxytocin. So what this means is that we have oxytocin released from the posterior pituitary gland. Now, when I say pituitary gland, the pituitary gland split up into 2 halves, the anterior and the posterior. The anterior gives off the majority of the hormones. There's only 2 that come from the posterior, and that is one called antidiuretic hormone that helps you regulate your osmolality and your urine output, and then the other one is oxytocin. So oxytocin is one of the 2 that come from the posterior pituitary gland. And what happens is, I think, from my understanding, is as a baby is moving down the vaginal canal, it will stretch the receptors in the vaginal canal. And as it begins to create this crowning effect or bulge, it will send out oxytocin to continue contracting behind the baby. I guess when women have contractions, I don't quite know when they say contractions what we're really talking about. I don't know if that's the uterine lining that is contracting. I'm assuming it is. And what happens is that when we have these quote unquote contractions, that oxytocin, the more oxytocin we have in the body from the baby crowning, gives off the more contractions that we have. All right. For some of you all who are mothers, you may have noticed that they want to measure your contractions, and all of a sudden they may be 4 minutes apart. Then all of a sudden they're 2 minutes apart, and then 30 seconds apart, and it kind of ramps up. That is the oxytocin in your body. That is the higher concentration of oxytocin. It will feed back onto the posterior pituitary gland, and because it's a positive feedback mechanism, that it will continue issuing more oxytocin, so you can get a full contraction out of the uterine lining. Now, there's another one, and I cannot think of it, but the big one is giving birth. Yeah, sure, Kendra, we'll definitely go over that. So, is there any questions? Is there any questions on positive and negative feedback? I'd probably just focus on that one, oxytocin. That's the one positive feedback that's the big deal. Negative feedback is pretty much going to be the majority of how hormones work in the body. So I guess we're okay there. All right, I'm on them. Okay. Yeah, you're welcome. All right, Kendra. So oxidative phosphorylation. Kiasha, that's how we manage stress. We don't want stress hanging out too long, right? And when I say stress, I mean cortisol. So we don't want a bunch of cortisol floating around for a long period of time because it'll mess our bodies up. So what happens is that's how the feedback system will come and bring cortisol back down to normal levels. Cortisol is always a little bit higher in the mornings when we wake up, just because that's the way things are. But eventually, as the day goes on, cortisol levels will drop off. And how they drop off is through this negative feedback system. The body says, okay, we have enough cortisol right now. We need to tone it back down. And this negative feedback system will go back to the hypothalamus and to the anterior pituitary gland to stop the beginning of the flow of releasing hormone and stimulating hormone, which are both found in the hypothalamus and anterior pituitary gland. Let me know if that clears up. Okay, cool. All right, so oxidative phosphorylation, back to Kendra's point. So I'm going to call it OP too, because it's a mouthful. This is metabolism. So when we have metabolism, we basically break everything down into glucose, right? Glucose is the monosaccharide that can enter into metabolism, and we get ATP out of it. So we have glycolysis, right? We go through glycolysis. We net 2 ATP, adenosine triphosphates. We have 2. So now all of a sudden we make this molecule now at the end of glycolysis is called pyruvate. Pyruvate is now broken in. We actually make 2 pyruvates per 1 glucose. So this pyruvate molecule will now enter into the CTA cycle, otherwise known as the Krebs cycle, citric acid cycle, and Krebs cycle. And then we will then reduce a bunch of stuff and make electrons. And anyways, we make this molecule called NADH, and then all of a sudden we take all this NADH and we run it through the oxidative phosphorylation cycle or process. So the purpose of oxidative phosphorylation basically is to phosphorylate ADP, because we are low on energy, so we've pretty much expensed all of our ATP, and now we have ADP, adenosine diphosphate. So the phosphorylation part is basically getting another phosphate onto that ADP to make it ATP. So we have diphosphate, and then we go through the oxidative phosphorylation cycle, and all of a sudden what that does is that it puts a third phosphate back on. So all of a sudden we've been restored back to ATP. Okay, ATP, adenosine triphosphate. And how we do that is through the mitochondria, and we have all these different electrons that kind of bounce down these reactions that create enough energy to place a phosphate on a adenosine diphosphate, and we create energy, and that's how we do it. Is that kind of what you were getting at, Kendra? Or do we need to go more in depth, or is that good enough? Okay. Is there anything else that we need to go over with oxidative phosphorylation? Are we okay on glycolysis and CTA cycle? Okay, it means, I guess Darian says we're okay. Let's talk about real quick monosaccharides, disaccharides, and polysaccharides. Let's break it down. A saccharide is a sugar, okay? Poly means a lot of sugars together. So we have a lot of sugars in line. This would be like what happens when if we were to eat a bunch of donuts, okay? The other morning I had a bunch of donuts. If we were to eat a bunch of donuts, chances are we don't need all the sugar in those donuts, right? So what happens is, and this is where fat kind of comes into too, if we have too much sugar, the body can get rid of it by storing it, and they store it as glycogen, okay? Glycogen is basically a bunch of polysaccharides placed together. Yeah, you could say that, Karen. And what happens is glucose, I think, is a monosaccharide because it doesn't get broken down any further in the glucose realm. Like sucrose is a disaccharide. So we will break down sucrose one last time into glucose before we can stick it into the metabolism cycles. So glycogen is more considered a polysaccharide. Now, let's say we go and we starve ourselves. Now our body will take our glycogen stores and start using them. And what it does is that it will break down the glycogen all the way down into glucose. And that's what happens when we enter the metabolism cycles, that we have to get everything down to glucose so we can use it. And that is basically getting it down to what we call a monosaccharide. Like I said, sucrose is a disaccharide, maltose is a disaccharide. There's a few other ones that are skipping. Lactose is a disaccharide. That's made up of like glucose and something else. I cannot think of it. You all can probably tell me what lactose is made of if you cover that in lecture. I cannot think of it right now. But it's two monosaccharides that make up a disaccharide. Okay. So Kiasha, what do we think about that? I mean, that's like a little introduction to saccharides. A carbohydrate. Do we know what a carbohydrate is? That's the same thing as a pretty much of a polysaccharide or monosaccharide. When we talk about carbs, we're always talking about sugars. It's the same thing. Yeah, you could say that. Basically, it's stored sugar in a polysaccharide form because there's a lot of them. It's like a lot of glucoses stringed together. It's a bunch of glucoses that are packed in together. They make up a polysaccharide, otherwise known as glycogen. You have to get these vocabulary words straight because they can confuse you. So a lot of them mean the same thing. Like table sugar, like normal table sugar. Yeah, it's also stored in the muscles too, Karen. Mainly liver stores glycogen, and then your muscles also, because it's the reason why it goes back to the fight or flight concept. For muscles, if you were to like fly out of there or fight, like glycogen is a very quick source of energy. You can break down glycogen within like a second and to use it for ATP to power your muscles and either fight or flight. You know how it's supposed to be like that quick reaction people do when they get spooked. Glycogen in the muscles is like right there, ready to go, to get metabolized out very quickly and to be used for energy to carry out whatever role the muscles need to carry out. Okay. So let's see what else. Because don't we have an exam coming up in lecture? I mean, surely, gosh, we have exams coming up in lecture. We're coming up on the 4th week. Oh, you did last week. Okay. Well, all right. So I already missed it. Got quizzes Monday. Okay. So is there any other, we got about 20 minutes. Is there any more questions, any more concepts we want to hit? They're going to be on the quiz or I guess part of Unit 2 now? While you all are thinking, I'm going to talk about lab real quick. Lab, we're coming up on, I think we're going to be doing tissues this coming week. It's a fairly long lab. You really have to be able to identify what these tissues look like and what features are in these tissues. It's more of a longer lab than most. We're going to be getting it for you in-person people. We're going to be getting the microscopes back out. We'll be checking out the microscopes and all that. But it'll be all right. I think we do have a quiz 2 coming up. I think, Karen, I think the chemistry is like in the beginning, like chapter 2 and chapter 3, and then I think we kind of start digging into the physiology and anatomy part. Yeah, I think so. Lakeisha, if you want to come to mine, if you want to get hands-on with the microscope, you can come to mine. I got Monday and Wednesdays at 6:30. So just send me an email. Yeah, if you're interested in working with the microscope, we only have an X amount. Newtown campus, SEC. Yeah, 6:30. Yeah, send me an email so I know you're coming. Um, so I can try to accommodate you. I mean, it seems like we have a lot of. Last semester, nobody came in person, and now like everybody is. I'm not sure why. SEC 303. That's the room right there. Monday and Wednesday, 6:30. So if you want to get hands-on with a microscope and see what some of these tissues look like in person, you can try to buddy up with somebody. You're gonna have to wear a mask, obviously. Get your temperature checked in the lobby area, and then if you want to see what a microscope looks like, we could probably split you up. Okay, that's cool. Just let me know. Send me an email if you're serious about coming, so we can try to make room for you. All right. So what else? Those are going to be the tissues. I think allergies are like kicking in my nose. It's like bothering me. What else do we want to talk about? I want to talk about the tissues real quick. I talked about the tissues. That's coming up because I think in lab you're going to have your first lab exam probably the next week or so, 2 weeks. I got to check. So we'll go over that probably next week when it gets closer. Do we have any other lecture concepts that we want to go over? Kind of covered a lot today, but we still got some time left. Krebs cycle. All right. That is also known as I've always learned it as the CTA cycle, so don't let me confuse you. Yeah, it's like you'll see it on the syllabus, Kiasha. It's microscopy words, histology, which is tissues, and all that introduction into lab 1. But back to Meredith's point, the Krebs cycle, basically the purpose of the Krebs cycle is to take it pretty much sets up oxidative phosphorylation. And what it does is that it reduces, and I'll explain what this is when I talk about it. It reduces this molecule of NAD. We've probably seen NAD. I can't even pronounce it. Nicotine diamide something something. And what it does is basically it's an electron carrier. It carries electrons. It's all it does. And we want enough electrons ready to go before we get to oxidative phosphorylation. So the purpose is to take all these electrons out of glucose, because electrons is what helps like power these different steps to put this phosphorus back on, put this phosphate back on ADP. So we have to get these electrons out of glucose. And when we do that, we stick them on NAD and we make NADH. Okay. It's just the hydrogen. When we reduce it, we put a hydrogen on there. And the electron is in the hydrogen atom. And so all of a sudden, the point of the Krebs cycle is to take NAD plus, which is ready to accept an electron, because once it accepts the electron, it's no longer NAD plus. The plus is gone. It becomes NADH. And we make a lot of those. I don't know how many we make, but we make a lot of those. And we don't really make ATP as much. We just get more electrons loaded up on NADH. And when we do that, that NADH will go down to oxidative phosphorylation and will help get enough power and energy to make ATP. That's pretty much the basis of the Krebs cycle. Meredith, is there any other key words that you see in the Krebs cycle you don't quite understand? That's like an overall gist of what the Krebs cycle does. Okay, I didn't want to confuse you. Good. No problem. All right. Any other concepts we want to touch on for the quiz next week or Unit 2? All right. Well, while you guys are thinking, I'm going to go find the attendance form here, and we will. I'll get this attendance form out to you so you can get credit. Yeah, it's 10 minutes. It's what it's supposed to be. All right, so I just sent over the link for the attendance form. I'm going to hang around and let you guys fill that out, and let me know when you're done, and you're free to go. Unless you all have a quick concept you want to go over, we can always hang around. If you want to talk about something that just popped in your head, just let me know. No, Kendra, I don't think so. We're just going to be doing tissues this week. You got to do the quiz, obviously. The quiz is going to come up this week, what we did over last week. But there's nothing you have to fill out. There may be like different, if you follow the lab manual, you'll see when you look at these, I think it's going to ask you to like what you see. And then I'll probably kind of point out like I've done in the past. I'll point out what you need to look for, like goblet cells or vice versa, and what these tissues are. So we'll kind of touch on it when we get there, when we start looking at them. No problem. I think you put my name, Kiasha. Just put Sundays with Mr. Mattmiller. Oh yeah, Meredith, I will. Thank you for reminding me that. Guys, I put these on YouTube. If you want to watch these again, my students have really pressed me to find a way to help with accessibility. And so I'm now on YouTube, which is a shocker. I never thought I'd say that. So I'll throw it up, Meredith. I'll put it up on YouTube probably sometime, probably before 10. I'll throw it up. Well, thank you, Kiasha. Come back and we'll do more. That's how I got through school, Meredith, is recording and listening to everything, and I wrote it all down. That's how I got through all my academics. Oh, thank you, Sierra. Colton, look on Blackboard. It's going to tell you that 4-letter word on there. It's going to like Bio 137 19ZE or 19 something like that. Those digits, letters, that's going to be your course section. Yeah, maybe Darren told you. Darian might have told you. Oh, Lee, just Google like Mattmiller Bio 137. I don't know my channel, man. I'm such a novice at this. Um, Google like Mattmiller Bio 137. You'll see all the different ones on there. All right, thanks, Caleb. No problem. Yeah, yeah. If you filled out the form, let me know and enjoy the rest of your night. I think just put my name, Darian. I'm not sure. And then Megan, what was your question? What is Spotlight Session? This is the Spotlight set. These are Spotlight sessions. I'm not sure if that's what you mean or not. Sure, Karen. No problem. You could put Sundays with Mr. Mattmiller or Sunday evening, Mr. Mattmiller. I think that's good enough. Oh, Angela, maybe that's how you pronounce your name. Just put Mr. Mattmiller Sunday evening, 137. I don't think it matters. All right, Lorena. Thank you. Have a good one. All right, Lakeisha. Have a good week. Check in next time. We'll do some more fun stuff. No problem, Karen. Check back in with me whenever. I do 4 of these a week. All right, Darian. Have a good week. Stay dry.